Okay,
Go.
you're on.
My dopamine level is not that normal. So, why we study nicotine? Because smoking and related diseases, smoking-related diseases are major health problems in the United States and all over the world. This slide I borrowed from my to illustrate the point. Now, this is from the CDC and National Health Center statistics on the rate of prevalence of smoking in the United States. You can see we've done a long way. Okay, although there's some other things not that great. That gender gap here is narrowing down, and in fact, some recent data suggests it's probably the woman's rate prevalence rate is going higher than men. What is also worrying is that it seems that we'll be flattening the curve here. We're just not able to go down more. Okay, although we have at least three different types of treatment available for us, we still hear flattening here. What is even more worrying is that with the new delivery devices of nicotine, such as e-cigarettes, dissolvable form of nicotine, the hookah, more and more younger people are, getting into these, using these devices. You wonder how this will affect, the rate of smoking all over the world. Okay, and I was coming here, and unfortunately, every day in the morning when I come, I see outside the faculty here at the university a lot of young people smoking. So this is where 90% of the smokers, adult smokers, started smoking when they were young. In fact, over 70% when they were age of 13. So, as Marina told you, in the beginning, right in the beginning, everything started with this nicotinic receptors. Okay? On the dominant history of tobacco plant, this is where it stops. She also told you, she also had a note, all the pharmacotherapy available to us now, such as nicotine replacement, different forms of nicotine replacement, varenicline, bupropion, all of them share in common that they target these receptors, nicotinic receptors, obviously in a different approaches from agonists, partial agonists, antagonists, and they have other properties too, which we're not going to talk about. So that shows the importance of starting and targeting this, the initial, where things started, the nicotinic receptors. She also told you that nicotine dependence in general and smoking addiction is a complex behavior, very complex behavior. And probably this arise from this widespread of multiple nicotinic receptors in the brain, in real relevant brain region. I'm not going to get into the details of it, but you can see the different subunits form the different subtypes of nicotinic receptors. They're distributed widely. Some of them are restricted. Some of them are more widespread. So it makes our life very complicated to understand which receptor is doing what, and can we get these specific ligands to target these receptors. This is another example. For example, here in the VDA, you can see the possible subtypes here. So you start asking yourself questions like, which subtype is doing what? Which aspect of nicotine dependence? Is targeting one subtype enough? Should we target more than one subtype in getting to understand what's going on in the big picture of nicotine addictions? Now, when you study addiction, in particular nicotine, for example, and smoking, it's a complex behavior. Many aspects need to keep in mind. There's always this initial experience and effect, initial effect of the drugs, and the alcohol field is very rich in that. So when you smoke, there's so many effects that goes from changes in body temperature, some sort of arousal, decrease in anxiety, memory enhancement. All these effects could contribute in this initial effect of nicotine, and later on into transition from simple use to regular use to dependence. There's also this issue of tolerance, the development of tolerance. It's not extremely well studied in the case of nicotine, like opiates, for example, but it does contribute. You probably know that Nobody starts smoking a pack a day overnight. The average, time lapse between, your first initial intake and you stabilizing certain level of intake is around 3 years it takes to go from the couple cigarettes to, this half a pack, or 2 packs a day. There's all the story of cue conditionings. I don't know if Marina got into a lot of details on that, but it seems that in the case of smoking, it's more prevalent than other drugs that use, and I'll show you a slide on that. There's also this whole aspect of the negative side, what George Kuhn called the dark side of the whole thing, the withdrawal, which, there's a lot of failures around it. That drives a lot of the relapse to nicotine and cigarettes. So it's a crucial part of the problem. And the whole relapse things, she just showed you, your fellow students, some data on a test, I would call it a test, to really get an understanding of reinstatement, which thought to model, some aspect of drug relapse. This is, for example, to illustrate the issue of cues. This is a study by Eric Doney when he was a postdoc. When the study took people who were regular smoker and switched them and gave them nicotinized cigarettes, regular cigarettes, and also another group gave them denicotinized cigarettes. Although it says denicotinized, but it still has a very low level of nicotine, 0.05 milligram or something. So you can see here, the group that got the regular cigarettes were able to sustain their intake. Okay. However, the group who just were giving, the denicotinized cigarettes, while they decreased a little bit their intake, but they were able to sustain it. In fact, with time they would stop, but you can see the power or the impact of the cues involved into this smoking behavior, from the smell, from the irritation, when people smoke, the fact that if you put in the cigarettes between your lips for years and years, the environment around you, all these cues has an impact. And nicotine seems to work, and although by itself is a relatively weak reinforcer, it enhances these cues. So all these aspects, are important. I know Marina talked to you about nicotine reward and reinforcement, and I want to focus most of my talk today on the other aspect, the negative aspect, the aversive aspect of smoking dependence, nicotine withdrawal. Okay? This is a famous, little bit old, but really well-known slide for Neil Banovich, where he tried to model early on what he called nicotine addiction cycle. That's not my definition. But you can see here, the lower line is a line below it. You start having a nicotine withdrawal. The upper line, above it, you can start feeling the subjective positive effect of smoking and nicotine. And it's a cycle during the day. You can see smokers start increasing their intake of nicotine throughout the day, and then when they go to sleep, they get into this zone of abstinence zone, smoke, withdrawal. The first thing they do in the morning is they smoke a cigarette, and in fact it's an important phenotype because it's the most rewarding, cigarettes of the day. And that phenotype is used in human studies and human genetic studies. In fact, it seems that it's most rewarding because with the nicotine intakes, increasing, they build up some sort of tolerance to that, initial effect, arouser effect of nicotine, the initial one. And at the end of the day here, the only thing they're probably doing is to avoid the withdrawal, the negative effect, by taking nicotine. So it's some sort of a cycle. So nicotine withdrawal is defined by many signs. Look at these here, from depression, insomnia, anxiety, difficulty concentrating, irritability. I put a craving here, increased appetite and weight gain. This is a very noticeable withdrawal sign. You can see here craving here is said added in DSM-5. In fact, it's a funny story. I don't know how psychiatrists sometimes do things off the record. It was in DSM-3, then it was taken in DSM-4, and now we're put back in DSM-5. Okay, I don't know why craving was not considered a withdrawal sign. It's a powerful, one of the most resistant withdrawal signs we have in smoking dependence. Now, this is, most of these symptoms and signs, except for craving, they peak a couple days after you stop smoking, and they'll go up to 2-4 weeks, after abstinence, and they play a major role in relapse. This is a questionnaire to give you an idea, on what do we ask a smoker who going into a withdrawal, an abstinence, post-cigarettes abstinence, what are the signs? And, this is, you can score that and use that in human and genetic studies. You can see here, I put a D, meaning we can discuss a little bit. If you look at those, which one do you think we can, in a way, model in the animals? Look at that and ask yourself. Suppose you came to the lab and heart rate. You can certainly measure that. This is pretty much a peripheral effect. You can measure that. That's important because in order for you to be able to study mechanisms and possibly targeting medication, you're going to be able to at least have a model in the animal that is relevant to what we see in real life in clinics. You can see here, how do you measure the urges to smoke, for example? How do you model that? As an example, headache, how do you measure that? How do you measure nausea and vomiting in rodents? Can you? No, okay? Although there's now people using gasping, the behavior in the rats, has a proxy model for vomiting and nausea. So some of them are amenable to, measure. Some of them are not. So what people in the field did is they divided these different signs to make it easy on us into three main aspects. Okay, the somatics, all the GI systems, sleep disorder, irritability, aggressivity, headaches, increase in food intake. All of them, in a way, are classified under somatic. These could be peripheral and or central mediated effects. They play a role, but not as big as a role, for example, of affective disorders such as anxiety behavior, depression-like behaviors, and others. And notice, and you probably know that when I talk about the animal part, I talk about anxiety-like behavior or depression-like behavior. You can't really have a depressed mouse or anxious rat. You measure, behavior that could, in a way, mimic that a little bit. The other part is the cognitive. Smokers who stop smoking, they have difficulty in learning, paying focusing attentions. All of these play a crucial role into a relapse. So how do we model this in the animal? Well, people are back and forth, about this. How do you give nicotine to start with? How long? And what's the dose of nicotine? All of these are crucial questions. So I want to have a little bit of discussion here. Where do you start when you decide on these things? Your advisor came to you and said, I want you to establish a model of radical dependence or withdrawal of nicotine. Nobody has done it. Where do you start? And what kind of information you need? Come on, I've seen you before in action. Nobody has to tell me. You're a great group. Where do you start? What kind of question you ask first? When you're trying to model this in the animal. Yes,
the
sir.
animal used by itself.
Well, let's focus on withdrawal now. Okay? Because these two mechanisms, reinforcement and withdrawal, could be separate. So what do you need to know?
If there is a rewarding effect of the drug.
You want to have some relevant to human signs, symptoms, right? Think pharmacology a little bit, okay? I know. The dose. What is important to know about the dose in order to establish a dose in this animal model? What kind of basic information we need to know? The half-life of the drug, the metabolism of the drug. The question should be immediately, is there species differences in nicotine metabolism and kinetics between human and my animal model? Okay? Immediate differences is that in the animal, for example, take the mouse. The mouse half-life of nicotine, guess what? It's between 5 to 10 minutes. So 5 half-lives, that's what it takes to eliminate over 90% of the drugs of the body. You know, then you got a problem. If you don't give enough nicotine in the mouse, you're not gonna see withdrawal, okay? The mouse is a better metabolic model for nicotine than the rats. And that's a huge issue when you develop drugs. For example, you've heard of bupropion, Zyban. Zyban is an antidepressant, in fact, okay? And it was developed after the fact, you know, for smoking dependent. And guess what? The metabolism of Zyban, of bupropion, is much closer to human in the mouse than in the rats. In fact, the rats is a lousy model to study the effect of bupropion, because most of bupropion in a human is metabolized to hydroxybupropion. That's not the case in the rat. So these are basic pharmacological and kinetics information that you need to establish the model. Second question would be, how are we going to give the drug? Now we resolve the dose. We can measure the level of the drug, and after giving to the animal, it gives us an idea. How do we resolve this route of administration? How do we give the drug?
It looks like you're giving him a pump.
There is mini pumps here, osmotic mini pump. There is inhalation. There is a drinking water, believe it or not, and there is the good old injections give to the animal. Who would go for an injection, chronic injection? And why? Who would not go with a chronic injection? Okay, let's make it simple. Why? Well, great. Half-life is very short. What else? Well, why not, you know, let's say because of the half-life, I'm going to give 5 injections a day. Who stops me from doing that? Me. Why? Well, yeah, but the more important issue is just this. You know, although you get the spike with the injection, which is good, okay, but it's so stressful. How do you separate the stress effect on a daily, regular basis, several times a day, you know, from this is, so it's the least preferred, you know, methods. What's the problem with the mini pump?
You have to do surgery.
Yeah, initial surgery and then recover fast. What's the main problem with it? It doesn't give you this fluctuation, right? It's just sustained, stable, but it's controlled. You know what you're getting, okay? In a way, and it's just clean, and you put the animals, you don't stress them or anything. They just live their life with nicotine, all right? How about this drinking water? Who doesn't like that, and why? I'm gonna put saccharin, okay? I have a controlled saccharin without nicotine, so who cares? What's the main issue with drinking water?
You control the dose that you have.
Why?
Because it depends on the mouse. No?
They won't taste. How will you regulate it?
They will drink it. If you put saccharin, they'll drink it. And if you keep the dose low enough, they'll drink it.
They can self-administer.
They will self-administer, you know, obviously with a very narrow range.
But it depends to the food, how much they take food, and then become thirsty and taking the water.
Well, I mean, nicotine decreases food intake, you know, but
So
there's a tolerance that develops.
Decreasing the thirst.
Well, there's a tolerance that develops to it with time. My main issue is what she said. Is just, I don't know how much each animal, or you can say, oh, I can measure that. I can measure the plasma level and get an idea, but the problem is nicotine has a very high first pass hepatic effect when given orally. That's one of the reasons oral nicotine is not, you know, now, is not considered till now a viable option as a nicotine replacement therapy. very high per first pass hepatic effect. Do we all know what's first pass hepatic effect?
What is going to this?
It goes to the liver before it goes to the general circulation. Most of it will be destroyed. Okay. Now the other is inhalation. Hey, this is probably the most the administration that has the most face validity, right? You know, you give them like, you know, smokers do. They inhale, right? That's why I envy so much the people in the alcohol field. It's so much easier to model the drinking. Mouse, depending obviously on the strain and the background, do drink alcohol, okay? So it has technical issues, you know, it's not easy to set, and it's not, surprisingly, not a whole lot is being done in the field. In fact, the number of paper use inhalation has a way to, you know, study withdrawal is very limited, probably less than 10 papers. Okay, most people use between those two, and till now, the mechanistic studies and some of the pharmacology suggest it doesn't really make a difference. Okay. This is an example of what you get. Here's some of our work here. This is in the plus MACE test. You can see that animals in withdrawal, they have a decrease in the time they spend in the open arms, indicating an anxiety-like behavior. Julie Brandy's group at UPenn used the NIH test, the novelty-induced hyperphagia, to show that the animals take a longer time to approach a new food that usually in their home cage they approach normally. This is, for example, the average, I don't know, I can't see well here, let me see. Yeah, average latency, that's hyperalgesia. Nicotine is in a human, have some analgesic property. So when you withdraw from nicotine, the general rule is withdrawal effects are the opposite of the initial effect. Okay? In general, you can develop hyperalgesia simply by using a hot plate. Okay? This is the type of somatic signs that I talked about a little bit, irritability. You know, obviously you can't see those in a human well. They start developing cold tremor. Yeah, some of it is close to the opiate withdrawal. Cold tremor, backing. A mouse or rat usually don't walk backward, you know, but they do here. Body tremor. You know, it's mild, like the human, withdrawal. It's not dramatic. They don't jump. It's not like opiate. Morphine withdrawal, naloxone precipitate morphine withdrawal is huge. Okay. Here, using aversion, you can use the condition-place preference that you heard about, a little bit. You can use it to measure aversion. Here also, for example, percent freezing. You can, in the hippocampal dependent, test, you can measure, there's nicotine going to withdrawal, there's a decrease of percent freezing to really study the effect on learning and memory. And also here, for example, ICSS used by Paul Kenny and Attila Marcoux and others, where you can see an increase in threshold in this, primary rewarding, test used in rat and mice. So you can model some of these, withdrawal signs that I told you about, and they're very relevant. And people started doing that. Now, first question we asked is, what kind of receptors, nicotine receptors, are involved in this? What are the subtypes, and where in the brain that mediate these withdrawal? We want to know the neural substrate, the mechanism involved, so we can develop drugs that target these subtypes that could help into the treatment. There's many of those. There's a lot of those. There's the homomeric, there's the hetero, there's so much different subtypes. There's the alpha-4 beta-2 by itself. And this alpha-4 beta-2, I'm not sure if Maria told you, depending on the stoichiometry, you have different, possibilities. You have the 2 alpha-4, 3 beta-2, or you can have 3 alpha-4, 2 beta-2. One is so-called high affinity, one is called low affinity. You got the alpha-6 containing receptors that may have beta-3, some others don't have beta-3 in them. There is also the alpha-4 beta-2 alpha-5, and then there's the alpha-3 beta-4. Although a lot of it is in the ganglia, but there's some of it in the specific brain region that has alpha 5, with or without alpha 5. So our life is complicated, and we're still unraveling, trying to discover which receptor subtype mediate which, sign. Keeps us busy a little bit. Multiple approaches were used. I am personally not happy with all these approaches, although myself use a lot of those. Okay. What's the problem with pharmacological approaches that we have in the nicotine field? If anyone comes to you and claim that they have a very specific and selective nicotinic agonist or antagonist or ligand, that is given systemically, especially non-alpha 7, you tell them goodbye. Okay? The only real selective tool or some sort of selective tool we have are peptide isolated from natural resources, toxins by Mike McIntosh, University of Utah, which were a huge advance in the field. But for now, we don't have really, like all these drugs that we use to treat nicotine dependence, nicotine, varenicline, they're dirty drugs. So that's a big challenge, okay? Unlike many in the field of other drugs abuse, like the opiates, which simply don't have those. Transgenic mice, knockouts, knock-ins. You'll be surprised to know that most of these studies that I'm going to show you in the field were done with newly mutant animals, not with conditioned knockout. I don't know of any studies published for withdrawal where they knock down in a specific manner a nicotinic subtype, nicotinic subunits in a specific brain region. Believe it or not. Okay? Not a single study published till now. Okay, there is some people trying to do that. The genetic approach. I'll speak about it a little bit. This is a early on work by Al Collins and Mike Marks at the University of Colorado Boulder, where they started this whole mouse genetics field for nicotine, but unfortunately very few people carried on. So all the advances and all the nice work done in the alcohol field does not really translate to the nicotine field. That's one area that I'm personally focusing more and more on. So we have a lot of challenges. Now let's see. I divided this for you in order not to play games. That somatic, affective, and cognitive. All right? What do we know till now? That the somatic signs of withdrawal are mediated by multiple subtypes. Not surprising. Okay? These somatics, some of them are peripheral, some of them are, central, so there's multiple signs of those, so I'm not surprised. This is, for example, here, let me see here. This is the somatic signs in beta 2 knockout. You can see here that beta 2 wild type here, nice increase in somatic signs. In the knockout, there is no significant decrease, right? This beta 2 subunits that Marina told you plays an essential role in nicotine reward and reinforcement, right? Well, it does not play any significant role in nicotine somatic sign withdrawal. And then for years, the field said, hey, beta 2 only play a role in reward and reinforcement, not withdrawal. Not true, because if you do the affective signs, here in the plus maze, you can see the anxiety-like behavior is totally gone in the knockout. People have repeated that using different tests. And also this is in the ICSS. When you give a relatively selective beta 2 antagonist, the hydroxychloroquine, you can elevate the threshold, ICSS threshold. There is involvement of beta 2 also in the cognitive part of these withdrawal signs, and alpha 7. Not surprisingly, because alpha 7, a lot of it is expressed in these brain regions involved in conditioning and learning. Cortical, the prefrontal cortex, hippocampus, amygdala, there's plenty of alpha 7 in hippocampus. So Jean-Pierre Cholinergic tried in this review to map these signs. Okay, that was a couple of years ago. Did a fairly good job. They told you about it, but I wanted to focus on this cluster of 3 genes on chromosome 15, the alpha 4, alpha 3, beta 4, and alpha 5. Okay? If it wasn't for the human genetic studies, that story would still be like any other story in a way, in a little bit of darkness. So this is a really what I consider a seminal work from Mariella De Biasi at UPenn now. She was at Baylor. She moved last year at UPenn, where she's the, in a way, the first to start doing these, brain studies. Okay, try to understand that where in the brain, what brain region mediate these cells. So she started with somatic science. And she went simple. She said, I'm going to inject a nicotinic antagonist, non-selective one, methamphetamine, in an animal random dependent on nicotine. For at least 2 weeks, they've taken nicotine. And then I'm going to try to precipitate these somatic signs and to see which brain region does this. So she injected in the cortex, hippocampus, in the nucleus VTA, and so no precipitation or withdrawal. However, when she injected this in the medial habenula and the IPN, she started a significant precipitation of withdrawal. So that immediately brought to the front this idea that the medial habenula IPN pathway plays an important role in nicotine withdrawal, at least here somatic signs. Okay. Now this mecamelamine that I told you about here, meca, it's a dirty drug, dirty antagonist, but it has a preferential affinity to alpha 3 beta 4. Now it's a virtual effect, not affinity, it's a non-competitive antagonist. So so that started us and others to think along that alpha 3 beta 4 alpha 5 may be an important subtype for nicotine withdrawal. This is a study, for example, that shows here by Mariella that in the beta 4 knockout, all nicotine withdrawal somatic signs are gone compared to the wild type. In our lab, alpha 5 knockout, there's a reduction of nicotine withdrawal. And using a fairly selective peptide antagonist for alpha 3 beta 4, giving ICV, we've shown that it's in a decrease and block, reverse totally nicotine withdrawal in a dose-related manner. So it's fairly well established now that when it comes to somatic signs, alpha 3 beta 4, alpha 5 cluster plays an important role in somatic signs of nicotine withdrawal. And most probably, this is through this medial habenula IPN pathway. Yes, sir.
So these somatic signs, are they the same as opioids, or how?
They're similar in nature, but not the same. They're certainly milder. And for example, have you seen a mouse going into morphine withdrawal? I mean, it's dramatic. They start jumping like popcorn jumping, right? They have diarrhea. They start to have some convulsion depending on the dose. You don't see that in the nicotine. You see a lot of cold tremor, backing, body tremor type, maybe, in the rats they're closer to opiate a little bit, but the intensity is certainly not the same at all. You can see that here, that it seems that this subtype plays an important role. Now, yes?
Is there a higher expression of alpha 3 beta 4 in the habenula or in the...
That's where we're coming from. So, no doubt, that is the distribution or the mapping of nicotine receptor subtypes, gives us a good case. Alpha 3 beta 4 really present in a very restricted manner in the brain, mainly in the medial habenula and in the IPN. So that helps us. This does not mean that other brain regions don't have it, but most of it is there. Same thing with alpha 5. Alpha 5 is present throughout the brain, but there is a lot of it in this brain region. So immediately people started saying, well, alpha 3 beta 4 alpha 5 is a crucial subtype to target for nicotine somatic signs, nicotine withdrawal somatic signs. So this paper just came out in General Science by Mariella, that I told you about Mariella De Biasi. Nice work that did the first time that's been done with nicotine, recorded nicotinic currents in the medial habenula. Okay, you can see and nicotine facilitate that, and that facilitation of nicotine is gone in the alpha 5 knockout. It's not, but it's still, that facilitation still exists in the beta 4 knockout and in the partially in the beta 2 knockout. Which is surprising. This is, you will see later, the behavior seems to suggest that these, this cluster in a way does not behave and has a cluster when it comes to pharmacology and behavior, because alpha 3 beta 4 seem to be separate from alpha 5. What Mariella did also suggest by using NK1 and 3 antagonists that this system seems to block nicotine-induced facilitation of excitability in the medial habenula. Adding that this brain region is really plays an important role, in nicotine with traumatic sound withdrawal, and seems to be also modulated by many systems in the medial habenula, including GABA and substance P, NK receptors. What we've shown also that while the alpha 3 beta 4, I told you, blocks nicotine withdrawal, it still blocks it, in alpha 5 knockout. So showing that, probably it's separate mechanisms involved and not one subtype. We still don't know what's the subtype that mediates this somatic sign of withdrawal in that specific brain region. Okay, the data till now suggests that alpha 3 beta 4 subtypes is one, and an alpha 5 containing subtypes, maybe it has a beta 2, we don't know, is involved. Okay, that's important because how Marina told you, a lot of genetics, the human genetic studies suggest that variants, especially an alpha 5 variant and genetic variant and alpha 3 beta 4 genetic variant, seems to be associated with an increased risk for smoking, especially in alpha 5, a mutation in the coding regions, lends to the expression of a less active alpha 5 containing receptors. That could explain what Kenny showed, an increase in nicotine intake using self-administration when he knocked down alpha 5 in the medial habenula IPN brain region. You can see here using self-administration and ICSS. We also showed it in the alpha 5 knockout, where nicotine is an aversive drug. When you do CPP with nicotine, the dose response curve is very narrow, very narrow. You move fast from being so-called rewarding to aversive in a very narrow, inverted U-shaped curve. While in the alpha 5 knockout, the aversive effect of nicotine is gone, and you can go to very high doses of nicotine, and you still see preference. So suggesting all of this suggests that that brain region and these receptor subtypes, alpha 3 beta 4, alpha 5, play a crucial role in smoking dependence. In one hand, decreasing or blocking the function of alpha 5 type receptors enhances nicotine reward and decreases nicotine withdrawal intensity. Some sort of I would say a cycle here, that could explain why in the human studies, genetic studies, there is a higher risk to move from regular smoker to heavy smoker when you have a deficiency or mutation in this alpha 5, because you'll find the drug more rewarding and less aversive. Okay, so you tend to take more of it.
So wait a minute, you confused me there. So if you take away the alpha 5,
You
then-
decrease the function, yeah.
then you don't, then you do self-administration.
You enhance the
don't,
intake.
don't have any aversive effect at all, so they just.
Well, they enhance the intake.
Yeah.
They found it more rewarding, more reinforcing.
Straight away then.
What do you mean?
So does that happen straight away? I mean, so they start self-administering, but there's no.
Well, at lower dose of nicotine, you're not going to find much difference
Right.
between the knockdown. When you start going into higher, and this probably because of the decrease of the aversive effect of nicotine. That region play an important role in aversion control. Yes?
I don't know if I'm going to get into this. You kind of mentioned a little bit about it, but does that mean that the alpha 5, the neurons expressing alpha 5 are different beta 4, alpha 3 neurons? Is that
I think there's differences in the expression. There's a paper by what's his name, Eric Turner, came out last year on that difference, the expression that suggests that there could be dissociated alpha 3 beta 4 and alpha 5. So, how about the upregulation? Marina told me about nicotinic receptors upregulation, right? These receptors desensitize, at least the alpha 4 beta 2, in this case, and then with chronic exposure to nicotine in the animal, in cell expressing systems, there's an increase of nicotine receptors levels, right? What is interesting, yesterday's talk by Pierre, when he was telling you, hey, if you see it across species, that's good. That's a good thing, right? Well, here we see it in human, in rats, and in mice. Since the 80s, this phenomenon is being described. In fact, the field invested over 30 years of resources and studies and work trying to understand why these receptors, upregulate. It doesn't make sense from a pure pharmacological concept. You give an agonist, you upregulate, instead of downregulate. Usually it's the opposite. You give an antagonist, chronically, you upregulate. Why upregulate? Trying to link desensitization to upregulation. Many studies, and Marina did probably summarize what the mechanism, but most importantly, is this relevant? Does this have any physiological, pharmacological behavior relevance? I have to tell you, the data till now, it's just not clear. It's not clear what this upregulation means and does. There is some studies, and I put it here, mainly correlation, that suggests that the time course of this upregulation after smoking abstinence may correlate or may be parallel to some of the withdrawal signs. This is a work, for example, by Tom Gould at Temple University, where he mapped out the upregulation time course of alpha 4 beta 2 in the hippocampus. You can see almost here 144 hours is gone, and in a similar group of animals, same condition, he also measured the percent freezing of nicotine withdrawal, and he concluded there is a parallel, time course. That's suggestive. Same thing as Julie Blandis recently said, okay, I'm gonna try to take two nicotinic partial agonists. One does not upregulate nicotinic receptors after chronic treatment. One does. Here, for example, Cellular does not, the partial agonist that does not upregulate, and Varenicline is a nicotinic partial agonist, which is the Chantix, does upregulate. One reverses anxiety withdrawal, and one does not. Cellular reverses the anxiety induced by nicotine withdrawal, Varenicline does not. This is a work by Kelly Postgrove at Yale, where she studied the time course using imaging of nicotinic receptors availability using a radioligand, an alpha beta 2 radioligand. You can see here, one day after smoking, there is not a lot of signal because there is still nicotine on board, displacing the radioligand. And then after that, it starts getting up and up, and then it goes down again. Because this is showing upregulation, and upregulation is phasing out. She did in the same study measuring measures of craving and withdrawal, and showed that there's some sort of a correlation in the time course. That's all what we know till now. Does it play a role, pose a role? We just don't know. How about other drugs of abuse? Do these nicotinic receptors, especially the one we talked about, play a role in withdrawal in other drugs of abuse? And this is a recent work that we did with people here, groups here in this campus here, Mara and our friend Raphael, where we studied opiates withdrawal, somatic signs of opiate withdrawal. You can see here, for example, giving ICV, the alpha 3 beta 4 antagonist totally eliminate morphine somatic sign withdrawal, the physical dependence, in a mouse line that overexpressed alpha 3, beta 4, alpha 5. You can see a enhancement in intensity of morphine withdrawal. The second one here, beta 4 knockout. Mariella did morphine withdrawal. There's a reduction, and there's also a reduction in the alpha 5 knockouts. What is more interesting is that in the same study, San Chen in Ken Kendall's group in my university showed there is a variant in this gene cluster, alpha 3, beta 4, that are associated with opiate dependence and opiate withdrawal, in human studies. So it seems that this most probably in the middle habenula IPN brain regions play an important role not only in nicotine withdrawal but maybe in opiate withdrawal also. This is a work it's brand new, not published yet. Doing THC, cannabinoids, withdrawal. You can, in the animal, you can precipitate somatic signs quite easily when you give THC chronically. You can see here when we gave the alpha 3 beta 4 antagonist, centrally we reduces nicotine withdrawal. THC withdrawal. That THC withdrawal is also reduced in the alpha 5 knockout. What is interesting is that when we tested those THC withdrawal in the alpha 6 knockout, there's a reduction. And when we gave a brand new selective alpha 6 beta 4, and that particular subtypes is really present in areas like the locus ceruleus, and other brain region involved in the opiate withdrawal and THC withdrawal, you can see an almost total reversal of THC withdrawal. Same thing, we did some human studies, genetic studies, and we found variant of alpha 6 and alpha 3 beta 4 seem to be associated with THC withdrawal, marijuana withdrawal also. So to summarize, these receptors seems to play a role not only in nicotine withdrawal but in withdrawal of other drugs. All right, any questions on these very complicated nicotine withdrawal and nicotine receptors and their involvement in the withdrawal? You can see immediately the field is lacking tremendous work on the cognitive signs, the affective signs. I mean, just lack of studies that needs to be done urgently, needs to be done. But the take-home message, everything to do with beta 2 seems to be involved itself with affective, cognitive type, and alpha 7 probably much more was the cognitive side for withdrawal, and alpha 3, beta 4, alpha 5 into this whole area of somatic signs. Yes?
Excuse me, sir. According to this upregulation and downregulation, we see that some people, some smokers, which they are not really smokers and keep, for example, smoking 2 or 3 cigarettes per day.
Mm-hmm.
And they are absolutely fine with this and they really enjoy it.
Mm-hmm.
But also we can see these heavy smokers that they just firing up a cigarette, they get 2 puff of it and then they just,
We call those chippers. Yes, they're social smokers in a way. In fact, they can stop anytime they want. They're not heavy smokers, probably not heavily dependent, and they can stop. They probably stop for a couple months, and then when there's a party and other people smoking, they smoke and they stop after that. Has not been studied in the animals. Very little studies also, to model this in the animal. It just, I don't know. This is one of the issues, in the field that's not being studied well. Good remark. Yes, sir.
So do these data begin to suggest that maybe it's the cholinergic system that's regulating the withdrawal? And is there any data on that?
Regulating nicotine withdrawal, you mean?
Withdrawal effects of drugs
It seems that
across systems, right? And there's no reason cannabinoids binding
no,
nicotinic receptors.
nicotine, these nicotinic ligands do not bind to, although endogenous cannabinoids such as anandamide and 2-AG are allosteric modulators of nicotinic receptors. And anandamide at fairly physiological concentration and levels, So, there's a work done, but all this was done in vitro in the expression system, This, especially the alpha 3, beta 4, alpha 5, data do suggest that it's more widespread as far as involvement than simply smoking and nicotine. Right. Yes, ma'am.
Is there an increase in the cholinergic tone during withdrawal, apart from the
Marine have done some work on that. There is, it seems to be an increase in cholinergic tones in some brain region, but these things not work very well. There is more studies. I'll show you some data on dopaminergic and some sort of cholinergic. There is an increase, yes. Yes.
Can social smokers in constant stress be addicted to?
I'm not a human pharmacologist, so I can't really tell you what studies being done. Does stress impact the intensity of withdrawal? Possibly. It has not been studied in the animal. No doubt that stress is an important mechanism involved in relapse. And you can see that in the animal in self-administration. You can reinstate, in the CPP and self-administration, nicotine intake and preference by simply what she showed before, the swimming test, or by a shock, mild shock, okay? So it could be, but no one have done it in withdrawal. Yes.
This has been seen an effect of THC over nicotinic receptor, because normally when people both mix nicotine with cannabis, so maybe if there is something kind of
Yeah, this is an early work done by Raphael, in fact. He studied the effect of THC on nicotine withdrawal and the effect of nicotine on THC withdrawal. This is, in fact, what led our work to start asking if those two drugs modulate each other withdrawal, what is the mechanism of this modulation? THC enhances nicotine preference, and THC, decreases. I wish Raphael was here, but I think he showed that THC reduces nicotine withdrawal intensity. So,
by regulating the nicotinic receptors, maybe.
I don't know if it's just THC. It's a very lipophilic substance, and it does not, to my knowledge, modulate even in allosteric fashion nicotinic receptors. Okay, but I haven't talked about that. When you block these enzymes that are responsible for degrading, 2-AG, the main endogenous cannabinoids, by doing MAG lipase inhibitor or in the knockout, you do reduce nicotine withdrawal tremendously. There was another, yes.
There's like how much of withdrawal in both in human brain and rodent brain and monkey brain is what Cook calls the dark side of addiction, but it was primarily amygdala and the force rising up in during withdrawal. Do you see folks how much of withdrawal become alpha 3 knockout?
The problem with alpha 3 knockout, they're not viable. They die within 24 hours, have major problems. Not surprisingly, alpha 3 is present in ganglia. Yeah, so the only way is you have to knock down alpha 3 in the specific brain region, which has not been done, surprisingly. I know Paul is doing some of the work on nicotine intake. But it's a good question. I'll show you some work on the stress, involvement of some stress system in nicotine withdrawal. George, Coop, and others have done some of the work. Yes, sir.
Are there any Bible or hybrid lines which are undeveloped which have different types of characters, like one is antagonist and another is agonist?
Well, varenicline in a way, a partial agonist is a mixture of the whole idea of varenicline didn't come from the vacuum. It's not like Pfizer jumped and said we have a great idea here. It came from the work of Jed Rose at Duke University, where he mixed nicotine patches with mecamylamine and saw a reduction in smoking behavior. So varenicline is a partial agonist. In the presence of full agonist, it becomes an antagonist. Most partial agonists are like that. Okay, but it's a very low efficacy partial agonist. Right? The problem with varenicline is that it's nasty side effect because it's a full agonist on alpha 3 beta 4. That's why you see one of the main side effects of varenicline is nausea and GI, okay, and cardiovascular, and it's a full agonist on alpha 7. All right, so and it's an antagonist of 5-HT3 receptors. It's a dirty drug. So that's why sazedine, for example, that I showed here, is another partial agonist that is more selective than varenicline. Yes, ma'am. I'm gonna take this one and then continue, and then we'll still can stop.
There are some data which I don't know if I believe them or not well, but I mean they show that exercise might decrease the intensity of nicotine withdrawal.
Yes, yeah, you have to believe those who are done in the human and in the animal. I'm not sure specifically on withdrawal, but in dependence in general. No doubt that exercise is used and will help into smoking cessation. No doubt about that. There's several papers on that in the human, and there's several papers now and getting in the animals, and I think what's her name at the University of Virginia have published a couple of papers in the rat on IV self-administration. Yes, I think that's an area. There's what's the mechanisms? Are these due to release of opiates, endogenous opiates? Because increased motivation. These are important points, and not to. I think you should start believing that a little bit. But there's nothing has been done on specifically on withdrawal in the animal. So how about other systems? This is a complicated slide. Shows that many systems, many neurosubstrates could modulate nicotine withdrawal, and people are starting gradually, doing that. The most important one that's being paid attention particularly, and rightly so, is the changes in dopamine signal. Okay, here this is the work by John Danny and Mariella. John is at UPenn now. Really nice work recently showed that in animals given nicotine chronically, and in this case it was in a drinking water. Okay, you can see there is a decrease in the basal nicotine levels by microdose and Nicholas accomplished in the mouse. This is, for example, 4 weeks of exposure and 1 day withdrawal, and then by the day 5 of taking nicotine out, they return to basal. Okay, and then here 12 weeks exposure, so longer, which is nice in study. He did 2 time exposure, 4 weeks and 12 weeks. It stays longer, up to 10 days to return back to basal levels. He also measures the tonic and phasic stimulation, evoke dopamine release in slices, which is nice. And he also saw a decrease in the 12 weeks, 1 day withdrawal in the firing of dopamine, phasic and tonic dopamine release into the slices. So there is a decrease in the mesolimbic system of the dopaminergic signaling. Okay, it's not only nicotine. This has been shown with other drugs abuse, such as cocaine and opiates. Okay, so and people have done it in the rat. You can see here in the rat giving mecamylamine dependent rat. There is an increase accumbens. There is a decrease in the dopamine release. Also, it is thought that this decrease could be responsible of some of the somatic signs and the anhedonia, aversion that we see in the animal and the human. Okay. Interestingly, in the prefrontal cortex, there is an increase of dopamine release. So, and we know that in stress and aversion, that increased dopamine plays an important role, and that's why people speculated that could be responsible, partly at least, in the anxiety and aversion that we see in the animal in going through withdrawal. So that's interesting. This is also work we've done and other people done in Ohio we started asking the questions. One of the mechanisms involved into regulating dopamine release, there's multiple mechanisms, GABA, glutamate, is the kappa system. And I know Charles Chachman is supposed to talk to you about kappa tomorrow, I think. You will see very interesting system. Well, so we asked the question, maybe there is this dumping, this decrease in dopaminergic signaling because there's an upregulation or a hyperfunction of the kappa signaling during nicotine withdrawal. And this study by Isola years ago showed there is an increase in prodynorphin message in many brain regions, suggesting there's an increase in the dynorphin activity. And indeed, when we injected, when we tested in the mouse, dependent mouse, nicotine-dependent mice, for example, here in the anxiety-like behavior, a kappa antagonist totally reverses, nicotine anxiety withdrawal, and the somatic signs are totally decreased, suggesting that there is an overstimulation of the kappa signaling in nicotine withdrawal. In fact, we've done that with other also, kappa antagonists, JD-TIC. The problem was these, JD-TIC went into phase 1 and then was stopped, phase 1 clinical trials. The problem was these are long acting. When I say long, they're long acting. Days and days in the mouse. So in the human even longer. So there's the new short-acting kappa antagonist, and in our hand they work well also in reversing nicotine withdrawal. So that's an area that deserves more work. Now, Marina did talk to you also about that the mistake that we do in the nicotine field, that we assume that nicotine is the only important substance. Well, there's over 4,000 different substances in this smoke and the tobacco leaf. Some of them seem to be playing an important role, have been shown in nicotine reward, and also recently a paper by George Cook and some of his collaborators shown, for example, that when they inhibit MOA, the monoamine oxidase, and you can see this is an aversion that I conditionate aversion. Focus on this the vehicle, it's almost gone, by day, let's see, 98 here, and you can see with the TCP and PLZ, they stay longer and longer and longer. So it seems that inhibition of MOA prolong the adverse effect of nicotine withdrawal. Not surprisingly, in the tobacco smoke, there are substances that inhibits monoamine oxidase. Most probably these substances, beta-carboline, harmine, and norharmine, that have shown also, by Francis Leslie at UC Irvine, that it enhances nicotine reinforcement. So that's something to pay attention. That's what should push us in the field to do more and more this smoking exposure to rodents in order to study the mechanism of withdrawal. Yes, sir.
Every time someone talks about the other 4,000 to 6,000 things, they always seem to focus on these beta carbonates. Is anyone looking at anything else?
Well, people have looked at acetaldehyde also. I mean, Francis Leslie did show that adolescent rat, for example, when they were when given acetaldehyde, they can acquire something sticky fast. Okay, and, but you're right. You just where do you start? That's the thing,
Excuse me, sir.
Uh-huh.
So can we say here that by decreasing of the B enzyme, the tremor which is coming after withdrawal, referring to decreasing this B, and then affecting more adrenaline and noradrenaline on the receptor, because it can stay more in the synaptic cleft, and also the metabolism of these catecholamines decreasing.
Can you restate your question a little bit differently, if you don't mind?
Here we can see that the MAO be decreasing. Am I right? During the
Inhibition, yeah.
Okay. Then, when somebody stops smoking, they will get a tremor. Am I right?
Yeah, to a certain extent. It's hard
Okay.
to see really
So
tremor.
can we say that, because from the other hand, when they are smoking, the blocking of some pathway keep them calm. And now they don't smoke. The concentration of the catecholamines in the synaptic cleft stay high.
During
And
smoking.
During not stopping the smoke.
No, I don't know if this is a, this is a, in fact, a slide by, Nora Volkov years ago. This is not during withdrawal. This is a smoke. No, I don't think anyone have done the MOA activity in the animal and human during withdrawal. So I don't know what happened with, for the activity of the enzyme during the nicotine withdrawal. Oh, so one of you asked about the HPA axis. In fact, this is a work that started recently. Started in George Kuhl's labs, and then Adri Brinshield's at now University of Florida, where he's really doing nice work, showing, for example, here that using the specific CRF1 antagonist in the amygdala, he shows that that dose-dependently decreases the anhedonia, shown he's using the ICSS in nicotine withdrawal, and also using a specific CRF2 antagonist, there is no effect, suggesting that CRF1 receptors may mediate some of these effective signs of nicotine withdrawal. Okay, and he's doing really nice studies on that, trying to understand the brain substrate and other peptides involved in stress also. So keep this in mind. Let me try to end it on this. I don't know how we're doing on time here, so we're doing fine. We can talk a bit more. Using this genetic approach, into studying some of the nicotine behavior, withdrawal, or reward. So a couple of years ago, I started getting involved because of my association with some people in the alcohol field, where I was amazed by the wealth of genetic approaches in the mouse into studying a lot of nicotine, a lot of alcohol behavior, and being familiar with Al Collins' works and his colleagues on, the inbred strain model work, beautiful work they've done with nicotine. I started asking myself, can we start using some of these approaches to study nicotine behavior and dependence? So one of the approaches I focus on is recombinant mice. And the idea was, in order to be fully translational, the animal and the human, work need to talk to each other. So there's plenty of GWAS studies in the human of nicotine smoking. Okay, there's issues in the phenotypes used. No doubt about that. I'm not here to discuss it. But overall, that field generated a lot of possible variants and candidates, right? And as you know, most of the outcome of these studies are correlation. You need to validate those in a model organisms. That model organism could be rodents, could be other models organisms too. And so it's important that to validate those in the animals. The reverse is also true. The animal models, at least the mouse genetic approaches could yield, and I'll show you examples, could yield a new, target that we've never thought of before. I'll give you an example of galanin receptors. Nobody thought that this system would be involved in nicotine withdrawal at all. So it's a two-way street, and that's what led us into, thinking about that. So one of the panel we use, and there's several recombinant inbred panel, is the BXD panel. Basically, it comes from, mating D2 and B6, B6 C57 black 6, and here pay attention to the substrain because there is many substrain of black 6 mice. There is the J from Jackson Lab, there's the N that comes from NIH and was giving to many different things. Now we're discovering that these substrains have tremendous phenotypic differences. You know, that's crucial because we can leverage these substrain phenotypic differences because they're really very similar to each other. You know, they come from the same parents. We can leverage these phenotypic differences in order to pinpoint, you know, specific, you know, candidate genes that could mediate these phenotypic differences. So the D2 and D6, and D2 and D6, as you probably know, they do differ in many aspects of alcohol and drug abuse. For example, the B6 are thought to be, you know, preferred drinkers of alcohol. The D2 are low preferred, you know, alcohol drinkers. They drink less alcohol. And same things in the D2, we hardly see any nicotine preference. While in C57, you can have a nice nicotine preference in the animal. So F1, then F2s, and they do sibling matings, brother-sister mating for 20 generations. And you can see there's now over 120 lines. Rob Williams at University of Memphis done a nice work on that. Over 120 lines, and you can see now the genomes coming from the parents is now clustered. And then in theory, if you have 1,000 lines and you can test the drug, you can go to the level of a gene that explain them some of the behavioral differences. So it's a powerful tool. What is more powerful that this database here, the gene network, contains over 2,000 phenotypic traits. Many of those are drug abuse related, cocaine, morphine, alcohol, and now we added the nicotine, and many other, you know, phenotypic data, you know, and also they're all sequenced. What else do you need? You can do the behavior studies. You don't have to sequence those mice, the F2s or whatever you have here. So all what you do is you add your data in the database, and you do all this beautiful analysis, and you can come up with many different things. So we started with around 20, 25, 30 strain, you know, lines, and we did simply nicotine withdrawal. And this is an example, the somatic signs. These are the different lines. This is the B6, this is the E2. There is not a whole lot difference in nicotine somatic signs, but you can see there is some sort of continuous trait, you know, here in the nicotine withdrawal, suggesting that, you know, there's multiple genes involved in that. Each one of them, you know, probably giving a little bit of peace here and there could explain this distribution. What is interesting is that you can use this database. You can say, get me the correlation in this brain region between, you know, this nicotine behavior and whatever data you have, expression data on any, you know, protein, any gene, any genes of any system. One of the things that came out very nicely is there's a negative correlation between nicotine somatic signs and the expression of galanine in the ventromedial brain area here. You can see very nice, and a positive correlation between the somatic signs and the galanine-1 receptor expression in the nucleus accumbens. No such expression was found, the correlation significant with anxiety-like behavior of nicotine. So now we have I would say a huge list of positive and negative significant correlation between a behavior trait in the mouse, relevant one, dependence, and many, many systems in the brain. So we can start saying, okay, you know, can we start validate those? And when we gave, oh my God, this is the Mac, this is Mac PC. Yeah. So this garbage here is simply showing that the effect of gallium. Gallium is a non-specific, or GAL1, GAL2, a non-specific agonist. Okay, and we can see that it reverses totally this nicotine somatic signs. Okay, so the data in the animals seem to suggest that this is a possible candidate for nicotine withdrawal. So we went and asked the clinicians, the human geneticists, do you see any genetic variants or genetic correlation, association between nicotine dependence and galamine 1 receptors? And indeed, this is what came out. Our study, and a study by Joe Cabells and Marina, came out in the same issue in neuropsychopharmacology, which made both of us extremely happy. That shows there's an association between variants of the GAL1 receptors and nicotine dependence. Using the FDND phenotypic scale. So that's the power of this. The problem is that the galanine field has its own problems, when it comes to ligand selectivity and coming up with simple selective antibodies for these different receptors. So, but that shows you the power of it. Now, any question on this before we move to the last presentation? That's what.
So your data suggests somatic signs are really important. I always thought the effective signs were going to be.
No, I didn't say that.
Well, no, you say it's related to smoking. So.
Well,
the effects.
The FDND does not see. That's an issue of human genetics. The FDND. What's the FDND? The Ferguson. Have you heard of FDND? So basically, it's a set of questionnaires that investigators use with smokers. For example, some of them are like, how long after you wake up you smoke a cigarette? Okay, that's a question that gives us an idea on, for example, on the when you wake up in the morning, this is the most important cigarette on withdrawal, for example. Or how important is the first cigarette of the day for you? Okay, do you feel this and that? Do you have any, for example, any type withdrawal signs? Do you feel good after smoking cigarettes? I don't have the list of all the questions, but these phenotypic data are put together in a questionnaire and a scale called FDND. There's other scale used, that's buried in it are nicotine withdrawal. The chief of them is how long after you wake up you go and smoke the first cigarettes. Okay, because if you care about the cigarettes, the first thing you do not fast enough is just go and seek a cigarette. So, having said that, I'm not. All what I'm saying is this subset of nicotine withdrawal, the somatic signs, seems to be indicative or play a role in the discovery of new targets. Now, people have thought that from a mechanistic perspective or conceptual perspective, these somatic signs they go fast, and we've shown that within a day or two they're gone. They play less role than the effective signs. Okay, I didn't show you the data because we're still working on it. We did not find any correlation with anxiety-like behavior in the galanine, but we found in many other systems correlation that we're working on. But your point is that don't underestimate what these somatic signs will give you. Okay. All right. So let's talk about FS7 a little bit and nicotine dependence. The reason I'm talking about this is that most of the work that pharma is working on when it comes to nicotinic, business is not smoking cessation, mostly. You'd be surprised. There were certain time or period they were working Abbott were working actively on getting an analgesic drug based on nicotinic, and some of our work helped in this. But mostly is, CNS-related disorders, specifically schizophrenia and cognitive, related disorders. And the focus of this effort is alpha-7 nicotinic receptors. These are very interesting subtypes. They play a role, tremendous role in learning and memory and in inflammation. Neuroinflammation and inflammation in general. There's nice body of work by Kevin Tracy and others that shows that there's an endogenous, pro-inflammatory system, anti-inflammatory system that is mediated by alpha-7 in macrophages. Very nice work. So, what do they play a role in dependence? Well, the early data, first of all, they're expressed in the right places. Marina told you they're expressed on the glutamate terminals in the VTA. They regulate dopamine release. They're involved. They're in the hippocampus, involved probably in cognitive signs of withdrawal. So we thought this is a great receptor subtypes that could play an important role. Well, so not really. The early data showed that the role in reinforcement is not that great and reward. Using the knockout, that's the data we published years ago. This is nicotine preference using 1 dose of nicotine 0.5. You can see there's no significant difference between the knockout and the wild type. When people at that time, the only selective, so-called selective, relatively selective alpha-7 antagonist, MLA, and you can administer it systemically, the data was all over the place. Some data giving MLA in the VTA directly showed a reduction of nicotine self-administration. Others giving systemically, there was no effect. The problem with MLA is also an alpha-6 antagonist. And when you block alpha-6, you block nicotine reinforcement. So it was not easy. So for years, people in the field didn't think a lot about alpha-7 and dependence, reward, and reinforcement. Okay? So some of my colleagues' work and my work shows that it is a target to pay attention to. Look at this, what Darlene Rosales and Michael McIntosh at the University of Utah did. So what they did is that using self-administration in the rats, PR schedule, when animals were giving in a nucleus accumbens shell, this is, let's see, which one? Oh, this is the antagonist. This is a selective nicotinic alpha-7 antagonist. It's a conotoxin, it's a peptide, so you have to give it by infusion directly in the brain. You can see here, when infused in two concentration in nucleus accumbens, there's an increase in the breaking point and the intake of nicotine. So you block alpha-7, you enhance, you increase reinforcement and motivation to self-administer nicotine. However, when you give an alpha-7 selective agonist, the PNU 28987, you see the opposite. So I don't know what's the mechanism, but it doesn't make sense, because when people knock down alpha-7, there's a decrease of nicotine-induced dopamine release in the alpha-7 knockout. So here, what she's showing that there is you block alpha-7, you enhance the reinforcement, you activate alpha-7, or you give an agonist, you block, reduce self-administration and nicotine reinforcement and motivation. That was very interesting. During the same time, we started getting into this the recombinant inbred panel. We said, hey, let's do nicotine preference, nicotine-based preference on these lines, and we can see also, this is a continuous trait here. As I said, suggesting the involvement of small effects genes there's a big difference between the DBA and the C57. One almost finds nicotine aversive, and the other one finds nicotine rewarding. So that suggests to us there's some role for different genes into this. So
Is that at all doses or just
this one dose.
That's the one dose.
One dose.
So
Gosh, if you wanna, yeah, I mean, imagine the work, the amount of
No,
work.
I understand.
But that's an important thing is, this is in a dose that in the C57 is causes preference. How about if we move to much lower dose or much higher dose? Probably we'll find different, when we do the mapping, we'll find different areas or loci in the chromosome that could mediate some of this behavior. And indeed, we found here, in the chromosome, chromosome 7 here, where the 7 genes is, we found, it's just expressing QTL, and also when we measured the expression level of alpha 7 in the D2 and the B6 in the nucleus accumbens, we found that the B6 have a higher level, significantly higher level of alpha 7 in the nucleus accumbens. So in a way, those DBA mice, they feel they don't show preference because they have a higher level of alpha 7 in the nucleus accumbens. So this led us to ask the question, is there any correlation using the WebQTL database between nicotine preference and other systems, the expression of other systems in the brain? So here, for example, nicotine preference correlate with a number of cholinergic neurons in the striatum. Okay. More importantly, is that here there is a negative correlation between the expression of alpha 7 in nucleus accumbens and nicotine CPP scores. So that's interesting. That suggests that when you activate alpha 7, you should see a decrease of nicotine preference. And indeed, so this is, for example, the PHA is a selective alpha 7 agonist. You can see the nice dose-related effect that blocks nicotine preference. Now pay attention to this. So this is the alpha 5, alpha 7 knockout. Remember what I showed you a couple slides ago? When we did nicotine preference, at 0.5 milligram, there was no difference between the wild type and knockout. So we replicated that. Now look what happened at a lower dose of nicotine. Lesson number 1. Don't go with one dose approach with pharmacological ligands. Never. Okay? If we have done our work more carefully at the beginning, we could have done a nicotine dose response curves, and we could have found that. Okay? And indeed, when we also use, this is alpha 7, when we use an alpha 7 knock-in, we get a function where the alpha 7 are hypersensitive. This comes from Mariella's and Baylor at the time. You can see nicotine preference is gone. So you activate the receptors, you block nicotine preference, you enhance the receptors, you block the receptors, you enhance it, you activate the receptors, you block nicotine preference. So what is interesting is that 2 different species, 2 different measures of nicotine reward, all saying the same thing, that maybe we need to start thinking about alpha 7 agonist as one approach for nicotine reward and nicotine dependence. Now, I'm not going to get into this a lot, but for just as a control, we did not see, I don't even know what is that. We did not see a change in cocaine preference. That's important, We've done also food. We've done lithium chloride-induced CPA. We didn't see a difference into, this is the 7 agonist. It does not block cocaine preference. So it seems to be fairly specific to nicotine. I am not going to get into this because it becomes very complicated, but using bioinformatics with my colleague, miles, he was able to find a gene network that involves alpha-7 in the nucleus accumbens. So what we did gene arrays on the nucleus accumbens from the alpha-7 knockout, alpha-7 wild type. We have a list of genes that differ between the nucleus accumbens, that differ between the two genotypes. We factor in the Ph.D. data, and we use several bioinformatic tools. And this network, gene network, that linked a lot of insulin, like insulin, signaling, insulin degradation enzymes and insulin receptors that are linked to nicotine preference in the nucleus accumbens. That shows you the power of some of these, tools. This would have never reported, never even thought of. If we want to really go investigate why going after insulin in the brain, insulin receptors or insulin protein, that are involved in signaling. So that's give us some work to do in the human and in the animal. With that, I'm going to acknowledge a lot of people. I can't go without acknowledging NIDA's funding for that, obviously, and the Virginia Foundation for Healthy Youth, for some of the work we've done on this, especially the genetics and also the work of adolescents. And let's see if we have questions, discussions. I'm all yours.
So I have a question about microglia activation. So
Oh.
it said alpha-7 activates microphages. Presumably also activates microglia, right? So
Alpha-7 agonist.
Yeah.
Yeah. So they decrease cytokine release, pro-inflammatory cytokine release.
So if you've got activated microglia, we're finding this a lot now, that seems to change the circuitry in the reward system.
Mm-hmm.
So my question, I guess, is that do you think any of your changes would be due to microglia activation as opposed to neuronal?
Possible. The other thing that I didn't tell you about is that I meant to show it is that what is emerging in the field of alpha-7 now, and this is the work of people like Roger Patke and others, and some of our behavioral work, is that alpha-7 receptors, it seems that exist in multiple conformations. One of them is a conducting state, that if channels opens and you get calcium is in, it's a very, very calcium permeable, subtype. And one of them, you don't have to activate and open the receptors to have an effect. You simply have, a bias type agonism involving, signaling, NF-kappa B in the case of inflammation. And recent work by an Italian group shows that, in fact, one of the possible way that an alpha-7 agonist could block nicotine reinforcement is that alpha-7 agonists increase the level in the nucleus accumbens in the mesolimbic dopamine system of OEA, PEA, which are endogenous activators of PPR alpha. PPR alpha activation, they showed, goes on and phosphorylate beta-2 nicotinic receptors, meaning a loss of function of beta-2, and that could explain why an alpha-7 agonist, really goes on and blocks nicotine reinforcement and reward. That's one possible explanation. The microglia one is a possibility. The other explanation, a signaling that is not related to really the to alpha-7 activation or conducting state. This is an interesting time for alpha-7. Yes, ma'am.
Where is exactly alpha-7 expressed within the nucleus accumbens?
It is. I don't know. I don't know where in the nucleus accumbens to be honest with you. I know in the VTA is on the glutamate terminals at alpha-7 state and regulate dopamine release. Yes.
Sorry, I've got two questions. First question is, could you elaborate a little bit more about possibility that those nicotinic receptors effects that you see modulating somatic withdrawal signs could be actually peripheral? And the second question is, what is the face validity of the withdrawal signs in mice corresponding to humans? And the question would be actually, what do people complain about? What is the most or yeah, what they complain during what they complain
Yeah.
about during withdrawal?
You just have to demolish all our work. That's all. These are very, very important questions, in fact. So let's start with your second question, and you can remind me the first question. What is a whole tremor has to do with anything to do with nicotine withdrawal and dependence in the mouse related to human, right? Well, probably from a face value, not a whole lot. Okay, it's an index of irritability, index of the animal is not feeling well. Okay, it's the same thing. The whole field of opiate withdrawal, nicotine dependence, is based on that. It's a tool. It's yeah, the writhing and all these things. It's a tool that we use that could means or not necessarily mean a lot in the human withdrawal experience and syndrome. Okay, no doubt that irritability and feeling irritated and not feeling well plays an important role. What is important also in human is the gain of weight that people have, the increase in appetite. Okay, there is little work is being done on this, believe it or not, and it's not necessarily
Column movement as well?
and the column move is not necessarily peripheral only. There is some work that's been done and shows that the involvement in the hypothalamus, signaling in this, increase of food intake after nicotine withdrawal. Not a whole lot is being done in the issue of colonic inflammations or colonic changes in transit, in nicotine withdrawal. Most of those are probably ganglionic and peripheral. Okay, not a lot is being done. All right, people in the field tend to not look at these peripheral, signs. Probably the focus is these affective and cognitive. But what driving us to look at the somatic signs is this beautiful data that's coming from Mariella and others on where in the brain and the possibility using mouse genetics to really tell us, give us new targets and new genes that we've never discovered before. So I wouldn't dismiss them totally, but at the same time, I agree with you, there's many other signs that probably are more important in a role in nicotine dependence. What was your first question?
I think you partially answered it. Peripheral effect versus central
Yeah. Somatic signs, some of them are peripheral, some of them are, central. And there's a simple way to do it. You inject a peripheral nicotinic antagonist in nicotine-dependent mice, and you can see there's some emergence of some of these somatic signs. But also people giving mecamylamine centrally in some brain region, and I showed Mariella's work that shows that you give it in the IPN and the medial habenula, you do precipitate somatic signs of withdrawal. Yes, ma'am.
You mentioned that also the glutamatergic system is involved in nicotine addiction. So I have two questions. How are these two systems interconnected, the nicotine one and the glutamatergic? And what do you think about the N-acetylcysteine treatment?
Let's start with the N-acetylcysteine treatment. There's a paper often cited by of Peter Kalibas and Tina Marcoux, nice paper in Biology of Psychiatry that shows that in a very small sample there's a decrease of number of cigarettes smoked when people, small group of smokers, were giving N-acetylcysteine. And N-acetylcysteine is not a clean drug in a way, but it does act on glutamate transporter, as you know. It seems some of the data in the paper, it shows that in glutamates re-establish the tone, the glutamate receptors, levels, seen decrease during nicotine withdrawal. In our hand, I don't have the data here. We also found an acetylcysteine does reverses several nicotine withdrawal. So, and I know there's some clinical trials planned or registered in the clinicaltrial.gov on acetylcysteine. Has suggested to you, discussed with you by Pierre yesterday. You ain't gonna see the light. This is an over-the-counter. There's a debate on the dose. Even there's some interesting data on cocaine addiction with acetylcysteine, but there's debate on the dose, low versus high. There's an issue with the high dose and toxicity, and there's no pharma company will ever goes and
no money.
Invest into this. So the better way is probably go after the target and try to understand clearly what's the role of these transporters in nicotine dependence and withdrawal. That's what led the main person working on glutamate system and nicotine withdrawal and nicotine reinforcement is Attila Marcoux, University of California, San Diego. She's done a lot of nice work on that. I didn't summarize it here, but she shows that some of these, glutamate receptors may be a target for modulation of nicotine withdrawal and nicotine reinforcement. It's just a lot of work out there for that team. Yes, ma'am.
Changes in alpha-7 receptor might modulate the cognitive deficits.
Might modulate, I'm sorry?
The cognitive deficits by
Yes. So this is an interesting question. By giving smokers an FS7 agonist, not only you'll be dumping down nicotine reinforcement and reward you've seen in the mouth, but also you will be reversing the cognitive deficit seen in nicotine withdrawal. So it's a double dipping, right? So that's an interesting. There is currently a clinical trial using some of these FS7 PAM. PAM is positive allosteric modulator, which a whole different area that I didn't talk about, for smoking cessation. That's a really good question, and I think the pharma is now gradually looking into this alpha-7 because alpha-7 agonists don't have many of the side effects seen with alpha-4 beta-2 agonists, such as varenicline and nicotine, that side cardiovascular, GI, nausea, vomiting. They don't have these effects, so that's one possibility. But, and there's several alpha-7 agonists now in phase 2, phase 3 trials for many, CNS diseases such as Alzheimer, schizophrenia, and cognitive deficit. So that's really an interesting area. Another issue is that how about if we combine, have a dual agonist, alpha-4 beta-2 and alpha-7. So that's the whole thing is still moving. Yes, sir.
So are any of these nicotinic receptors expressed in adipose tissue?
Yes, they are. There's nicotinic receptors everywhere. Everywhere. From the skins to the lungs, they're everywhere. Especially alpha-7, because I don't know if Marina told you, it's probably the oldest receptor in the from a evolutionary perspective is one of the oldest receptors, the oldest in the nicotine. So that's why it is expressed in many different tissues outside the CNS. What do they role? What they play their role? I don't know. We're interested in the pain and inflammation, and they do play an important role. I'm the one stopping you for going to lunch, right? Last question. Well, I'll be around today and tomorrow. If you have any questions or discussion, feel free. Thank you.